Hello everyone, welcome to my biology class, and in today's class we will be studying about translation. Remember, translation is the last step of central dogma. We are already done with transcription and also we have discussed the self-replication process. So I would recommend everyone, please watch the initial videos on the said processes and then come to this. And you must have knowledge of RNA before coming to this topic. So let's get started. Now, what is translation? You know that we have discussed in a video on RNA that how many types of RNAs are there, right? So you must have the knowledge of them because we would be discussing about all of these in the translation because they will play their role in this process. So when messenger RNA synthesizes the proteins, the process is known as translation. In NCERT, it is very short. I would strictly recommend to follow the video because I will be telling you in the steps. So let's get started. Translation, as I said, it will be happening in steps. Okay, so we will go with the steps one by one. First of all, the amino acids will get charged. Okay, amino acids will get charged. Now you know that the translation process, it is happening in the cytoplasm of the cell. So if a 1-marker question comes, what is the site of translation? Then you need to answer that it is cytoplasm. Now the first step is amino acids will get charged. You know that amino acids are there in the cytoplasm and they are 20 in number, right? Whenever we are talking about proteins, proteins are the polymers and amino acids are the monomers. So the monomers for this process, of course, they are going to be amino acids. So first of all, they are required to get charged, okay? Then only they will be charging transfer RNA. So here AA means amino acid, ATP means adenosine triphosphate, and there will be one more enzyme that will be required for this process, and this is amino acyl tRNA synthetase. Okay, it would be great if you could take your notebook and start writing the chemical reactions as it is proceeding in the video. Okay, so in this reaction we can see that amino acid is reacting with ATP and the enzyme name is amino acyl tRNA synthetase. This enzyme name is very important because they may ask you which enzyme is required for the activation of amino acid. One more thing you have to remember is for the activation of amino acids, we do require magnesium ions, okay? So this is our amino acid adenylate complex, okay? Our amino acid adenylate complex is formed, means our amino acids, they are being recharged now. The first step is done. We will proceed to the second step, and which brings this amino acid to the transfer RNA. And when this gets attached to the transfer RNA, then it is known as transfer RNA is charged. So the second step is the charging of tRNA. So while it is happening, we would be writing amino acid adenylate complex with the enzyme. This will be uniting with tRNA, and what will happen? This will attach to the transfer RNA and the remaining components will be set free. So this will be our AMP, it will be released, okay? So first step, amino acids will be charged, that we have done. The second step is the charging of tRNA. Remember, I said in the transcription process too that the translation process, it is not a one-step process. Okay, so there are various steps of it, and the first step for it is initiation. Then what happens is elongation. You can recollect because all these terms were told in the transcription video. And the last one, of course, will be the halting process that will bring an end to the translation. Okay, so first step, major step, will be initiation, then elongation, and then termination. So we are coming to our third step now, and third step, of course, will be our initiation. So I will discuss about prokaryotes and eukaryotes both, how it varies in them. So please stay focused. Now, initiation process will utilize some proteins as well, and these will be known as initiation factors. Okay, so IF means initiation factors. As I said, I will be talking about prokaryotes and eukaryotes both. So here we will be discussing initiation for both of these organisms. Remember, in prokaryotes, whenever we are talking about initiation factors, we do need to use this abbreviation IF, and it will be followed with a number, which initiation factor is going to help in the translation process. Just to remove the error, what we do for our is we use small e IF, means this will be the initiation factor of the eukaryote. Okay, and after IF we will be writing some number according to the process that is going to happen. Okay, so let's start with the initiation process. Now, if I'm talking about prokaryotes, you know that prokaryotes, they have 70 S type of ribosomes. Whereas if you talk about eukaryotes, then they have 80 S type of ribosomes. If I bifurcate them, then the larger unit in them is of 50 S and smaller unit is of 30 S. Whereas in the eukaryotic ribosome, the larger subunit is of 60 S and the smaller subunit is of 40 S. This is must to remember. Okay. Now, what happens in the initiation process? First of all, this smaller unit of the ribosomes. So if we are talking about prokaryotes, then of course the smaller subunit will be 30S only. Okay, it will unite with messenger RNA. Because messenger RNA, they have information in the form of triplets called codons. This information will be translated in the form of amino acids. Okay, so this smaller subunit or the 30S subunit will unite with messenger RNA, and during this process IF3 means initiation factor 3 is going to help. And what will happen? This smaller unit will get united with the messenger RNA. Okay, so 30S will be united to messenger RNA. If the same process would have happened in the eukaryotes, then in them the smaller unit is 40S and this will be uniting with messenger RNA, and in this process there is a little change that initiation factor 2 is going to help out. Remember, we are studying eukaryotic one, so that this name will become EIF2, means eukaryotic initiation factor 2 will help out in this process, and by this the smaller unit will be united to transfer RNA. Right? Once we are done with this step, means this is our messenger RNA, okay, and this is our 5 to 3 prime, okay. And one more thing I just want to tell you, the ribosome has 3 sites on it, okay, E site, P site and A site. Okay, remember you have to write all these sequences in a serial order. There should be no mistake. So E for exit, P for peptidyl site and A for amino acyl site. Okay, amino acyl site. Remember, the first amino acid will always come to peptidyl site. So this is also one of the important question of the board examination. First amino acid comes on which site of the ribosome? Answer is P site. Okay, so our messenger RNA is already there from 5 to 3 prime, right? And the smaller subunit has attached. Okay, and suppose this is the sequence like on RNA, and this is the sequence called AUG. Now this AUG, which is there on the messenger RNA, right, it will get positioned in such a way so that it lies close to the P site of the ribosome. This is very important. So once smaller subunit gets attached to messenger RNA, the messenger RNA is positioned in such a way so that the initiation codon or the initiator codon, which is AUG generally, or it could be GUG in some of the prokaryotic cases, it will be positioned close to the peptidyl site of the ribosome. Okay, once this is done, then the next step will happen. And one more thing, all the regions prior to the, means they are occurring just before the initiator codon, they are not going to help in the translation. Okay, so these are our untranslated region. I have talked about these UTRs in detail in the transfer RNA video. Do watch it out. Okay, so UTR is nothing but it is untranslated region, and it is the region that occurs prior to the initiator codon. Okay, now we are studying still initiation process, and it is going to be a lengthy one. See, the smaller subunit has already been attached, and in the case of prokaryotes it is this, and in the case of eukaryotes it is this one, which is encircled. Now the next job is of transfer RNA. You know that we have codon AUG. Now transfer RNA will bring the amino acid to AUG, and with genetic code table we already understood that AUG codes for methionine. So transfer RNA will bring methionine, right? And the amino acid, as it was told in step number 1, first the amino acid has to get charged, and in the second step it will be attached to transfer RNA and then it will be charged. So our transfer RNA got charged, and in the case of prokaryotes, the transfer RNA brings first amino acid as the formilated methionine. So we just write in short formyl met, means formilated methionine. But if we are talking about eukaryotes, the transfer RNA brings a simple methionine. So here the students who are preparing for the competition exams, this is a must to remember that in prokaryotes methionine is there, but it comes in the formilated form, whereas it doesn't happen in the eukaryotes. Okay, so this transfer RNA, suppose here again we'll be talking about 2 steps. So this is our prokaryotes, right? 30S was already united with the messenger RNA. Now this will unite with transfer RNA, okay, and that has brought formilated methionine. Okay, remember in this step which initiator factor has to happen? This is IF2. Now by this what will happen? The 30S was already united to the messenger RNA. Now this has also been attached to the formilated methionine bringing tRNA. Okay, so they all will be united together. Now if the same process would have happened in the eukaryotes, then how we would have written it? It would be 40S messenger RNA because the smaller subunit has attached to the messenger RNA, right? Now this will get attached to the transfer RNA that has brought the methionine. So this is the IF3 which is going to help in this process, and at the end of it, what is going to happen? All these components, they will be united together. As a result, 40S will be united to messenger RNA, and this will be attached to transfer RNA which has brought methionine. Right? So remember, this is the step which is happening in the prokaryotes, and this is the second step which is happening in the eukaryotes. So let's see in a diagram what has happened till now. Okay. Now this is our messenger RNA which we are going to draw, and suppose this is our sequence AUG. Okay. And this is the smaller subunit which has already been attached. And now what will happen? Our transfer RNA, suppose this is our transfer RNA, and this bottom part, you know that it is always going to be anticodon loop. Okay. Now loop is the Solon structure of the transfer RNA, and the back portion is known as the arm. So anticodon loop is a one which has anticodon, and this gets attached to the messenger RNA, right? So as per the base pairing rule, it will be U here, A here and C here. Okay, so this is what has already been happened, and this component AUG, initiator codon, it is there at the P site of the ribosome. Okay. Now we will come to the last step of the initiation, because we have to form the initiator complex. Once initiator complex is formed, that means now we can step in the elongation process. Okay. So let's start with the next step. Now we already had this 30S messenger RNA. This was attached to transfer RNA which was formilated methionine. Now our things are ready. Now the role of the larger subunit, it will come. Means this is the time to make the functional ribosome, and functional ribosome will only be formed when the smaller subunit will be united with the larger one. So that's what we are doing here. In this process, initiator factor 1 will help. So overall, as a result, what will be formed? 70S ribosome will be formed. It will be attached to messenger RNA along with transfer RNA formilated methionine. This is the process we have done in the prokaryotes, because here the subunits are 30S and 50S, and that's what that was told to you in the beginning only. Okay. Now let's see how it happens in the eukaryotes. Okay. So in the eukaryotes, this structure, smaller subunit was already attached with the messenger RNA, and this messenger RNA was attached to the methionine. So here also the right time has reached where the larger subunit will be uniting to the smaller one, and in this process eukaryotic initiation factor 1 will also help, and eukaryotic initiation factor 4 will also help, because of which the ribosome will become functional, because whenever we unite smaller with the larger one, we get the ATS type of ribosome, and this will be attached to messenger RNA, and this RNA has of course methionine in it. Okay. Now see, our initiation complex is being ready in the case of prokaryotes as well, and in the case of eukaryotes as well. Now stay tuned for elongation and termination in the next video. I request you to please subscribe my channel and share with everyone in your known ones, so that they can be benefited and they can learn their whole NCERT in a simplified way. Thank you.